From page 13 of your student guide, we've got one learning intention we're covering today in this PowerPoint. Under the heading of alternative splicing, we are going to explain how different proteins can be expressed from one gene. The learning we've covered up until now has included transcription and also translation being the most recent of our learning experiences. In translation, we looked at the role of tRNA, the role of the ribosomes, start codons, stop codons, and the fact that peptide bonds join amino acids together to form that polypeptide chain. Splicing is the process where we see introns that are referred to as being non-coding regions of the transcript being removed or being spliced out, while the exons that are regarded as coding regions get joined together to form a messenger RNA that has a continuous sequence of nucleotides. The modified mRNA, so the mRNA that is produced after the result of splicing, is called the mature transcript. And that mature transcript is what leaves the nucleus to head to the ribosome to be involved in translation. The diagram in front of you is showing chromosomal DNA and how alternative forms of splicing could lead to alternative proteins being produced. If we think about a primary transcript of messenger RNA, let's say for example in protein 1, being cut up and its exons being spliced together to form a molecule of mRNA ready for translation, what we see is that we could end up with one particular protein being made based on the exons that remain in the mature transcript. So if we look at protein 1, we've got a sequence of exons numbered 1 to 6. We've got introns represented by those light blue areas. But for protein 1, we see a splicing process taking place where we see all of the introns spliced out, but we also see exon 3 being spliced out. So that results in a mature transcript of exons 1, 2, 4, 5 and 6. That will see the translation of a very particular protein based on the genetic language 1, 2, 4, 5 and 6 are coding for. We can use the same chromosomal DNA though, using the same primary RNA transcript, but if you look to protein 2, under what we call alternative splicing, here we've seen all the introns spliced out, they always get spliced out, but this time we've also seen exon 2 and exon 4 almost treated like introns, so they get spliced out. So the mature transcript from this gene is now 1, 3, 5 and 6, and again it's going to produce a different protein. So this is what we refer to as alternative splicing. We've got one gene there represented by exon 1 to 6 with the intron areas. Through transcription we get the same primary RNA transcript 1 to 6 with the same introns. But the alternative splicing, i.e. the removal of some introns and some exons, you'll see that you can end up getting very different sequences in the mature transcript. So as a reminder, introns are always removed, but under alternative splicing, some exons can also be removed. Different proteins can be expressed from one gene as a result of alternative RNA splicing. So when we have that primary transcript, some exons may be retained, some may be treated like introns, and that will result in different mature transcripts being produced. I would ask you here to evaluate your own learning based on the single learning intention we've covered as shown on the screen.